Hi everyone, I'm going to present my research on a new method for a scalable large-scale DNA barcode classification. It's called PROTAX-GPU, done as part of my research internship. In terms of what I'll be covering, I'll first provide some of the bioinformatics background on DNA barcoding and the need for a scalable analysis pipeline. And I'll then explain my proposed approach, including the model and the optimizations which were done. And finally, I'll conclude with some experiments that validated on a large real-world barcoding dataset and demonstrated its improved computational performance over some previous methods without sacrificing accuracy. So first, I'll go over a bit of background on what is being worked on. So DNA barcoding is a genomic approach to species identification, and it has gained some traction over the past decades. So traditionally, we would have relied upon phenotypic differences, and that both takes a large amount of manpower and might only be effective up to a certain extent. For example, certain traits might only be present in specific life stages. So DNA barcoding instead relies upon leveraging genetic variation to discriminate between organisms. And this approach saves a large amount of work since all it requires is just the DNA sequence. And what I have here is a very high-level overview of the barcoding workflow. We start with an unknown organism that we wish to identify, extract its DNA, and sequence that. And typically, we only care about a standardized gene region since even the smallest genomes are in the order of 100,000 base pairs. And we finally use this sequence to identify the species by comparing it to a database of barcodes. So we're essentially sampling from a specific region within the genome for classification. But however, even with this enhanced throughput provided by this method, there's still a bottleneck which is present. So this graph compares the number of barcode sequences found in bold, which is one of the largest DNA barcode databases to date, against the number of reference sequences used in the deployment of various barcoding models over time. And what we see here is that the amount of data that we have still grows much faster than the models we have, which are able to utilize it. And ideally, we would want to incorporate the entire reference sequence database into a model. So as you can see, analyzing these ever-expanding data sets poses both a computational and a statistical challenge, since the larger databases tend to also contain a greater number of erroneous sequences or incorrect taxonomic labels. And as we all know, the use of GPUs in machine learning have enabled deeper and larger models, such as AlexNet, to be feasibly trained. And since DNA barcoding appears to be similarly compute-bound, GPU-accelerated barcode classification can potentially address this gap. So here I'm going to go more into detail about the problem that we're trying to solve. So we're given a query sequence of about 600 to 1,000 base pairs, as well as an existing linnean taxonomy. So that's the domain kingdom phylum system we all learned in high school, and a reference sequence database. And what we have is a classification problem. More specifically, we want to map the query to a position within the taxonomy, usually by comparing it against the stored database and the stored taxonomy we have. And to put things into perspective, the database can have up to 13 million DNA sequences as of now. And the data is also very high dimensional since we have around 600 to 1,000 base pairs per sequence. And if you want to look at it through the lens of a large language model, we need to have about 7 to 13 billion characters just in context, though in reality, only a very small subset of this dataset is actually relevant to making the prediction. And that brings me to PROTAX. So PROTAX is a probabilistic model for barcode classification, and recently reached 91% accuracy in classifying unseen arthropod sequences. And what it does is that it models the probability distribution over all taxa and decomposes it into probability starting from the top down. So in this diagram, you could see the root probability being decomposed as we step down in depth. And it's also able to account for undiscovered and unknown taxa, as well as estimate the fraction of mislabeled sequences in its predictions. And how it does this is that it uses a hierarchical series of Bayesian multinomial regression models that are able to incorporate predictors or outputs from other classifiers. And these are used to predict local conditional probabilities. So that's seen in the equation. It's the probability of the query being assigned to some node j given its parent. And we chain these local probabilities together to produce a global outcome probability. So in our case, we only use the sequence similarities of the k-nearest sequences within the taxa for our predictors. But in the most general case, the main idea of PROTAX is that it's a flexible framework for combining classifiers and features into a probabilistic output. And you can sort of think of it as a mix between k-nearest neighbors and softmax regression. It's ultimately a semi-parametric method that uses k-nearest neighbors to load the relevant features of the reference sequence database into the design matrix shown on the right, which is finally used for softmax regression. So in terms of drawbacks of PROTAX, the implementation of PROTAX currently executes only on the CPU, and it sequentially computes the sequence distances and probability decomposition. And as our dataset grows in size, the computational time taken by it increases proportionally to the number of entries that we contain in our database. And this linear growth in runtime quickly becomes prohibitive as the reference sequences are added every day. So although PROTAX already leverages some optimizations such as search space pruning, its runtime is still largely bound by the fact that it runs on the CPU. So to enable analysis on the more modern large-scale biological datasets, we're proposing a transition from a sequential to parallel computation. So to address this gap, we implemented PROTAX in JAX, which is a high-performance Python machine learning library. And it not only provides easy access to hardware accelerators such as GPUs and TPUs, it's also wrapped in a high-level interface and provides composable function transformations, which allow us to combine different classifier implementations in just a few lines of code. And because a key feature of PROTAX is that it can utilize different classifiers as input, the composability provided by JAX also makes it straightforward to experiment with integrating different classifier types into PROTAX. So here I'll quickly go over the inference steps in PROTAX-GPU. And in this figure, I have the computation graph of the model. The circles represent operations while the blocks represent intermediate representations of data which we're working on. So first, what it does is it computes the sequence distance between the query and reference database in parallel, computes the k-nearest sequences to the query within each taxon, and finally, it computes local probabilities and chains them together as an overall probability using a simple lookup. So in terms of the data representations it uses, we represent a has-a relationship between a node and a reference sequences with an entry of 1 inside of a sparse matrix. So this matrix has the shape of this matrix is the number of taxons present in the taxonomy, as well as the number of columns are the number of reference sequences we have in the database we want to compare it against. And the main reason we have this is that it allows us to apply our sequence distances to our taxa in just one multiplication. And additionally, we represent the nodes within the path starting from the root, also as a sparse matrix. And the reason for this is that we can represent the overall probability of an outcome as the product of the conditional probabilities. And if we pre-compute the paths, we're able to chain them together also in one matrix multiplication. And here I just have the mathematical justification for that earlier example I have here. So in summary, representing the taxonomy as a sparse matrix allows us to save both on memory and utilize the parallel architecture of GPUs compared to the hierarchical tree structure that's used in PROTAX CPU. And this matrix formulation also allows us to naturally map entries to grids of the GPU cores, and allows us to assign naturally assign threads to each row in the matrix. And that in turn defines the reference sequences the thread is allowed to operate on. And additionally, we also implemented k-nearest neighbors on sparse matrices as a custom CUDA kernel and dispatched it through a custom JAX operation. And the reason for this is that it's inefficient and impractical to implement k-nearest neighbors using high-level JAX functions. And the reason for this is that iterative algorithms and dynamic shapes aren't well suited for direct implementation in JAX. And secondly, as a custom operation, this allows our entire stack to be written in JAX and allows for JIT and PMAT compatibility, which offers huge performance gains and compatibility with multi-GPU systems. So here I'm showing the results of PROTAX-GPU on a single query. In terms of the experimental setup, I have 37,000 reference sequences in a database, and I'm using k-nearest neighbors with k equals 2, and I'm comparing it against the original C implementations of PROTAX. So C version 0 is sort of a baseline unoptimized version of PROTAX, and C version 2 is the CPU version of PROTAX with a couple of optimizations in place. And as you can see, the JAX implementation of PROTAX achieves single query performance of about 1.5 milliseconds, which is roughly 100 times faster than the CPU version of PROTAX. And additionally, I've evaluated PROTAX-GPU in terms of its performance and accuracy. So for this experimental setup, I've also evaluated it on 37,000 reference sequences, and the models are both parameterized with MCMC sampling. So the tables here show the performance of each model relative to taxonomic rank. That is, its performance at different depths within the taxonomic tree. And here we're evaluating the model in terms of accuracy and perplexity. So the accuracy is meant to measure the predictive performance of both models, while perplexity is to assess its uncertainty in each prediction. So here you can see that the accuracy and perplexity of the two models both match as we step down the taxonomic tree and achieve about a 97% species level accuracy. And as expected, perplexity starts to explode the further down we go in depth, since there's exponentially more outcomes at the species level compared to the genus or family. And finally, for this last experiment, I've compared the runtime of, I've evaluated the performance of PROTAX-GPU in terms of its runtime as we scale the reference sequence database up. And how I did this was I've created a synthetic taxonomy and both a reference sequence database and measured the single query performance over 10 runs at each reference database size. So it appears piecewise linear scaling in runtime. It's likely due to how JAX internally handles resources, but from 1.5 million sequences onwards, it scales linearly. So the main takeaway here is that the evaluation with 7 million reference sequences takes only about 3.1 milliseconds and only uses 4.2 gigabytes of VRAM. And that's well within the capabilities of modern consumer-grade GPUs, which are typically equipped with 8 to 48 gigs of VRAM. So in summary, what we've done is parallelized PROTAX to make use of GPUs and achieved about 100 times speed up without sacrificing any accuracy. With that said, there's still a few areas that could potentially benefit from further exploration. One potential area of improvement is that the redundancy can occur in barcodes assigned to higher taxonomic levels. For example, there may be some substantial overlap within barcode sequences usually used to identify orders or family and even genera. And this repetition might be unnecessary when our goal is simply just to assign a taxonomic label to a DNA barcode. Secondly is that PROTAX has a very limited number of learnable parameters. If you recall, its regression parameters are assigned to each rank in the taxonomy, and it only scales with the depth of the taxonomy and the number of predictors we have. So it's ultimately still just a shallow model that works well with reference sequences in the realm of 10,000 sequences, but might not naturally scale as well as we go past 10 million onwards. And finally, k-nearest neighbors is also sensitive to noise and the curse of dimensionality. And since our current barcode sequences are already 600 base pairs, the accuracy of k-NN highly depends on the quality of training data and the choice of k. So these problems might be further exacerbated as our dataset size and sequence length increases. So it's also expected to be an issue as the barcoding datasets continue to grow in size. And with that, that's the end of my presentation.
Hi, so that is amazing presentation. I just have a quick question on the results. Can you go back to the slide where were you showing the accuracy? Yeah, the previous, yeah, this one, yes. So my first question is that why exactly top at 1? And second thing is that about the perplexity, just I would like to know a little bit more, and what is a good score for this average perplexity, this one?
Yeah. So by top 1, I just mean I'm taking the maximum probability at each rank in the taxonomy and taking that as the prediction and comparing it against the ground truth label. And in terms of perplexity, it's you can think of it as exponentiated entropy. It's just a measure of uncertainty within the for a probability distribution, and these results are sorry, the results that are shown here are what I expect, since as we reach down to make these finer grained decisions, such as at the species level, there's a lot of probabilities which are almost zero.
Okay, got it. So for the accuracy top at 1, is it, I mean, it looks to me like equivalent to recall at 1. I mean, is there any difference? Any specific reason why exactly you mentioned here accuracy, not the recall, if it is at top at 1?
No, there isn't any specific reason I decided to call it accuracy. Yeah.
Okay, thank you. That's amazing presentation.
Hi, Roy. You said how the tool was using k-nearest neighbors PROTAX for on the CPU, but that couldn't be implemented in the GPU. I didn't quite catch what were you doing instead on the GPU instead of the k-nearest neighbors?
Oh yeah. So what was done is that I've implemented a parallel version of k-nearest neighbors, which operates within going back to this diagram here. It operates in parallel in terms of each node in this tree concurrently. And in the old implementation of PROTAX, it would compute k-nearest neighbors sequentially for each node.
Okay, so this is the change that you made.
Yeah.
Okay, got it. All right, thank you.